Welcome to this presentation on the use of RNA in vaccine development. I am Karine Breckpot, and the work that I will be presenting is from the Laboratory for Molecular and Cellular Therapy, which is located at the Vrije Universiteit Brussel. So what we do is we try to generate therapeutic cancer vaccines. And these therapeutic cancer vaccines are based on the knowledge that a cancer cell, which is depicted here in orange, is different from a healthy cell, which is depicted here in green, based on the fact that this cancer cell shows aberrations in its DNA, which ultimately lead to the expression of proteins, which we call tumor antigens, that are not shared by the healthy cells. And these tumor antigens, when they are presented on the cancer cell in MHC molecules, can be scanned by the T cell receptors of T cells. And when there's a match, so meaning that the T cell receptor binds the tumor antigen together with the MHC molecule, this tumor-specific T cell should be able to kill the cancer cell. Now, in our work, we are focusing on what we call neoantigens, or tumor-specific antigens. These are antigens that have not been encountered by our immune system prior to the development of these tumor cells. So they are considered non-self. And as a consequence, there is also no central tolerance against these neoantigens. So that means that any T cell with a T cell receptor that can bind these neoantigens should be able to do this with high affinity. Moreover, these T cells, they should be effector T cells and should not be hampered by any regulatory T cells that might occur during central tolerance development. Now, this makes neoantigens an ideal therapeutic target, but of course, these neoantigens are very specific for a particular patient, and we need to be able to identify them, and this today is still a bit of a challenge. What you need for that is to collect tissue, tumor tissue, and healthy tissue, for instance, peripheral blood cells. After isolating the nucleic acids out of these tissues, can then perform next generation sequencing and compare on the basis of genome, on the basis of the transcriptome, what actually are the differences between the nucleic acids found in these tumor biopsies versus the nucleic acids found in these peripheral blood cells. And this comparative sequencing is then fed into a computer algorithm, and you have multiple computer algorithms that you can use to predict which neoantigens are actually present in the tumor biopsy. Now, as I said, T cells are the cells that should be the ones that kill the cancer cells. But T cells that react to neoantigens should first be activated, and this activation needs to be done by professional antigen-presenting cells. And for that, in the laboratory, we focus on dendritic cells. Dendritic cells that were first described by the late Ralph Steinman, who you see here on this picture holding a dendritic cell. And these dendritic cells, when they are immature, they can actually take up antigens, and these antigens can then be processed into smaller peptides for presentation on these so-called MHC molecules. Now, such a dendritic cell, when it would interact with a T cell, would not be able to activate a T cell. For that, you need maturation of the dendritic cell, and this can be done by alarm signals, which then lead to a mature dendritic cell capable of presenting the peptide to different T cells and capable of providing the necessary stimulation to activate these T cells. And this T cell activation, when this happens, and the T cell is specific for the antigen that is being presented, in our case, a neoantigen, then this T cell should be able to go out of lymph nodes where this interaction with dendritic cells happens and go in search of the target cells, in our case, the cancer cells, to destroy them. Now, we use messenger RNA in this situation in two ways. First of all, we use messenger RNA to deliver the neoantigens to our dendritic cells. And secondly, we use messenger RNA to deliver the alarm signals to the dendritic cells. So let's focus for a minute on the messenger RNA that we use to deliver neoantigens. Neoantigens, as I said, can be multiple and are very specific for a patient. And as we are using prediction algorithms to find out which neoantigens could be present in the tumor, there's also a high need to test these neoantigens before we go into a real vaccine production. If you use mRNA for neoantigen delivery, and if you use the good old technique of using plasmid DNA to make this messenger RNA, you are confronted with a few issues. These issues are that this plasmid DNA preparation followed by the mRNA production is quite laborious and a multi-step processing that takes time. As you can see here, there are at least 4 different steps, starting with the production of your plasmid, in which you then insert your target sequence of interest, meaning one or multiple neoantigens. Then these plasmids need to be transformed into bacteria so that they can be copied to a billion of the exact same plasmids. And then after purification, these plasmids need to be restricted by enzymes. So you have a linear format, and then you can use it in a 4th instance to make the mRNA in vitro. Next to the fact that you have these multiple steps that are time-consuming, this also means that there's a risk of contamination with, for instance, bacterial contaminants. There can be leftover plasmid DNA, and if we start to grow bacteria, this is done in a system where you use antibiotics, there's also a risk of contamination with antibiotics. Now, these are inherent to the plasmid DNA template. Other risks are a little bit more inherent to the way that we use these plasmid DNA templates in the context of neoantigens. Since the cloning takes time, we bring in the sequence of different neoantigen epitopes in what we call a tandem minigene, meaning that we have different sequences that we link together with adapter sequences. And these adapter sequences actually can give positional effects. That means that if you change the order of your neoantigen epitopes, it could well be that certain epitopes are not expressed well, while others are. And vice versa, if you would change it again, that maybe the ones that were initially well expressed are not well expressed anymore. Moreover, a risk, and something that we have seen in the laboratory, is that these linker sequences that you use to really couple the different neoantigen sequences might become processed and presented to the immune system, making up what we could call nonsense epitopes that do not make any sense to be used by your immune system. And of course, something else that comes along with that is that it's a fixed vaccine composition. So if during analysis, it would turn out that one or multiple of the neoantigens included in the tandem minigene is actually not immunogenic, not eliciting a T cell response, you can actually not just simply take it out. You have to start over again with your plasmid preparation and go through the 4 steps that are shown here. And this for us was something that we wanted to circumvent. And to circumvent that, we thought of a synthetic way of making the DNA template, and in that regard have a limitation or a restriction of the number of steps that are needed, as well as avoidance of bacteria. So what we do is we actually take the sequence of a single neoantigen, which is shown here in green, and we couple that in an assembly PCR together with 2 oligonucleotides that have a fixed sequence and that actually contain all components to make a messenger RNA. Once this assembly PCR is done, we amplify the DNA molecule that is generated as such using a PCR again, so that we have sufficient amounts of material to then afterwards perform the in vitro transcription. What I should also mention is that our single neoantigen is coupled to what we call an HLA class II sorting signal. And this HLA class II sorting signal is actually the sorting signal of dendritic cell lysosomal associated membrane protein, which is a membrane protein that is depicted here, and that actually has a tyrosine-based signaling molecule, which is shown here in the red circle. And this tyrosine-based signaling molecule makes sure that the proteins that are coupled to this dendritic cell lysosomal associated membrane protein are actually transported to the lysosomes so that they can potentially be loaded onto MHC class II or HLA class II molecules. Now, why do we use that? Not only to make sure that we can include neoantigens that might elicit CD4 positive T cells, we also include it because we know that these sorting signals allow a much better presentation of peptides and epitopes that are bound to HLA class I. So overall, our system, the synthetic production, has a higher flexibility because each time we produce a template for one antigen, so we can mix and match afterwards in the vaccine. It is a bacteria-free system, and since it's only 3 steps, there's a reduction in the labor that needs to be performed, there's a reduction in time as a consequence of that, and this is a system that is prone to automation. So in the end, you can allow a robot to mix all of the components needed to generate the mRNA. Of course, making such a synthetic DNA template is one thing. Proving that the mRNA that derives from that is equal to the mRNA that you can derive from a plasmid DNA template is key. And to do that, we first started with an easy system where we used dendritic cells from donors that were HLA-A2 positive, which we then loaded via electroporation with mRNA encoding an antigen of interest. And then we co-cultured those with T cells, of which we made sure that they have the right T cell receptor, so meaning a T cell receptor that recognizes both the HLA-2 molecule as the antigen that we delivered under the form of mRNA. And then in first instance, we looked at interferon gamma production to see whether there's really presentation of the antigen and to what extent. As you can see here in the 3 graphs, we did this for 3 different antigens, GP100 and NY-ESO1, which are actually cancer testis antigens and differentiation antigens, and then for 1 mutation antigen, which was P53. What you can appreciate is that when T cells are co-cultured with dendritic cells that are electroporated with mRNA that encodes a totally different antigen, then we don't see any interferon gamma production by our T cells. Once we deliver the antigen of interest and the correct one to the dendritic cells, we see that both in the case where we produce the mRNA starting from our synthetic or our plasmid DNA template, that we do see production of interferon gamma, signifying that there's presentation. And in certain cases, it is the plasmid DNA template that performs a little bit better than the mRNA. In other cases, they perform equally well, and yet in other cases, it is the plasmid DNA template that performs a little bit worse than the synthetic DNA template. Overall, for these antigens that are presented in HLA-A2, we can say that at least with the synthetic DNA template, we can produce mRNA that allows the dendritic cell to present the correct peptide. As I mentioned, we also should be able to present epitopes in HLA class II using this dendritic cell lysosomal associated membrane protein. And for that, to prove that, we did a similar setup where we have dendritic cells that are positive for HLA-DP4, were modified to encode the antigen MHA3 using messenger RNA, and then co-cultured with T cell receptor positive T cells. And again, we looked at interferon gamma production next to the activation marker CD69 and OX40, showing that in both cases, when we use this mRNA produced with synthetic DNA template or a plasmid DNA template, we actually have good interferon gamma production or good T cell activation. To prove that this lump or this DC lump is really necessary to have this antigen presentation, we also had the MHA3 sequence without this DC lump. And in this case, the last bar here, you can see that there is hardly any presentation of the peptide. So this led us to test true neoantigens. And for that, we had a little bit of a different system in the sense that we selected for HLA-A2 presented neoantigens, at least predicted to be presented in HLA-A2, and we co-cultured these dendritic cells with CD8 positive T cells that have not yet have a T cell receptor. And so since we are stimulating T cells de novo, we added a mixture of mRNA to mature these dendritic cells. And that mixture is called trimix, where we have two molecules, CD40 ligand mRNA and constitutively active toll-like receptor 4 mRNA that should activate the dendritic cell, and then CD70 mRNA that should help give a signal to the T cells to proliferate. Again, we looked at interferon gamma to see whether our system worked. And as you can see here, with 3 different neoantigens that were evaluated in 3 different donors, we can perfectly see that some of them stimulate T cells in all 3 donors, like for instance, the DHX40B and the one here shown below, while some only elicit T cell receptors, responses in 2 out of 3 donors. So showing that there's indeed a need to test these neoantigens for their immunogenicity, and also showing that our platform is perfectly suited for that. Now, so far, we have used trimix mRNA as an adjuvant, but we wanted to improve our dendritic cells compared to this trimix, because with trimix, we still have quite a lot of interleukin-10, and that is a cytokine that hampers T cell stimulation. And also it hampers interleukin-12 production, and interleukin-12 is actually one of the factors that we need to stimulate good CD4 positive T helper 1 response and CD8 positive T cells. And so we decided to use the CD40 ligand and constitutive actively toll-like receptor 4 to activate the dendritic cells, and then to capture away the IL-10 that is produced at the time using a decoy IL-10 receptor, and to stimulate IL-12 production with interferon gamma. We compared this trimix adjuvant, as we call it, to our prior trimix adjuvant, looking at the transcriptome of dendritic cells that are modified like that. And we show here in the principal component analysis that actually dendritic cells modified with a green fluorescent protein, which shouldn't do anything with the dendritic cells, trimix or tetramix, are actually completely different in transcription profile. And here below, we show that 55 genes are actually upregulated in trimix modified dendritic cells, while they are downregulated in tetramix modified dendritic cells, and vice versa. 55 genes are downregulated in trimix and upregulated in tetramix. And about 330 genes were either in both downregulated or in both upregulated. We then wanted to have a clearer view on what this really signifies, and for that we did a gene ontology analysis. And what we observed is that actually delivery of trimix mRNA to dendritic cells gives cells that show pathway activation as you would expect in response to bacteria, while with the tetramix, what we see there is that our dendritic cells have a transcriptome that resembles very much what would happen when they encounter viruses. Then we looked at the number of genes and notified that in tetramix, most of them were more expressed than when we had our trimix dendritic cells, and we verified this on a protein level, looking, for instance, at interleukin-10, where we see that both are present in trimix and tetramix dendritic cells, but in tetramix, they shouldn't bother the cells because of the decoy receptor. We then looked at interleukin-12, which should be highly expressed in our tetramix dendritic cells, which was the case as shown here in the last bar. And then finally, we also looked at the number of co-stimulatory molecules, amongst which CD40, CD80, and CD83, which were all upregulated in our tetramix dendritic cells compared to our control, as well as compared to the trimix in case of CD40, CD80, and CD83. So this shows that these tetramix modified dendritic cells do have a good potential to stimulate T cells. And so that was the next thing we addressed using a similar system where we looked at HLA-A2 positive dendritic cells that were co-cultured with CD8 positive dendritic cells, and then we looked at the dextramer staining. The dextramer staining looks like this when you do the flow cytometry. And then when you summarize these data, we see that indeed tetramix dendritic cells are more prone to stimulate T cells. We then looked at the capacity of these T cells to kill tumor cells. And what you can appreciate here is that there's indeed a decline when tumor cells are co-cultured with T cells stimulated with trimix or tetramix dendritic cells, signifying that there is tumor cell killing over this 10-hour time lapse. This brings me to the conclusion of this work and the future perspectives. So we have chosen to use dendritic cells as a platform to kind of deliver our mRNA in vivo. And in that regard, we make a personalized vaccine already because the dendritic cells are autologous, of course. Our vaccine that we are producing is moreover personalized because we use neoantigen mRNA, which we deliver with our synthetic DNA template mRNA platform. And we use tetramix mRNA using our plasmid DNA templated mRNA platform. The neoantigens, we did not select them ourselves. For that, we collaborated with a company which is called MyNeo, and they have a special algorithm to do that, an engine which should allow us to really select the best neoantigens. And the idea is to use this in a colorectal cancer setting in the following months. So I hope in the next year to have data on that. So then the only thing that is left to do is to acknowledge everybody that worked on this, which are Emeritus Chris Tielemans and then the mRNA dendritic cell manufacturing team, as well as the 2 PhD students that worked on this project. Also the collaborators from MyNeo for the neoantigen prediction, and of course the funding agencies for the financial support. If there are any questions, I will be happy to address them.